In 2019, the FDA approved the first ever drug specifically targeted for postpartum depression, Brexanolone IV. And what's unique about this drug is that it's a synthetic version of an endogenous neurosteroid, Allopregnanolone. Researchers theorized and confirmed via clinical trials that by restoring third trimester levels in postpartum women, they could alleviate symptoms of depression. This research fascinated me. This link between mood and the unique physiology of the perinatal period. Personally, it impacted me because at this very time, after the birth of my second son, I was struggling with my own symptoms of postpartum anxiety. Professionally, this knowledge regarding allopregnanolone inspired my master's research, which is looking at allopregnanolone in relation to preterm birth, and which I look forward to sharing with you now.
Allopregnanolone, which I'll refer to as allo, is a metabolite of progesterone, and it's considered neuroactive for its ability to rapidly, within seconds, alter neuronal excitability. Allo modulates GABA by enhancing inhibitory neurotransmission. The result is sedative and anesthetic effects on the body. Because of allo's actions at GABA, it has important pharmacological implications, and it has been investigated in conditions ranging from Alzheimer's to autism. Allo is stress responsive, and in acute experiences of stress, allo levels rise to return the body back to homeostasis. During pregnancy, allo mimics progesterone, increasing steadily and peaking at term. The high levels of allo found late in pregnancy ensure positive birth outcomes, including timely parturition and proper neurological development of the fetus, by doing a number of things: dampening the maternal stress response, restraining oxytocin secretion, providing neuroprotection to the fetus, and contributing to uterine quiescence. Animal models demonstrate that in conditions of chronic stress, allo is lowered. Low allo late in gestation is associated with decreased gestational length and impaired cognitive and neuroendocrine function in offspring. In humans, low allo is associated with perinatal mood disorders. Separately, we know chronic stress is a risk factor for poor birth outcomes. This raises the question, is low allo during pregnancy associated with birth effects in humans? Well, our study focused on one poor birth outcome specifically: preterm birth. Preterm birth is birth prior to 37 weeks gestation, and it affects about 1 in 10 infants. Of those preterm infants, they comprise almost 80% of neonatal morbidity and mortality. Importantly, preterm birth is characterized by substantial racial and socioeconomic disparities. Our understanding of developmental biology tells us that the intrauterine environment is plastic, it's responsive, and that external environmental cues affect developmental trajectories, resulting in phenotypic variation. Maternal stress, which includes psychosocial stressors such as racism, discrimination, socioeconomic disparities, well, it's a key environmental cue, and with regards to allo, we're looking at how the maternal fetal placental neuroendocrine system may be affected. Is stress-responsive allo altering this neuroendocrine system, resulting in phenotypic variation, in this case preterm birth or an abbreviated gestational length? Well, that's exactly what we investigated. In a nested case control study using banked serum samples, we hypothesized that women who gave birth preterm would have significantly lower allo than women who gave birth at term. We investigated 2 time points in pregnancy: blood sample 1 and blood sample 2. And now I'll review our study methods. Feel free to pause your video to take a look at the details of this synthesis pathway. We determined maternal serum concentrations for analytes listed in gray, including allo and 5 other steroid hormones: allo's precursor progesterone, 2 additional metabolites, pregnanolone and epipregnanolone, as well as the stress hormones cortisol and cortisone, which are obviously a key aspect of the stress response system. The progesterone metabolites, seen here in the lower left-hand corner, are present in very low concentrations. They're also molecularly very similar, and therefore they require an assay with significant sensitivity and selectivity. In order to quantify these analytes in pregnancy, we developed a novel, validated, high-performance liquid chromatography tandem mass spectrometry assay. Feel free to pause your video again to get a better look at this slide. For the purposes of this presentation, all you need to know is that we included healthy women with singleton pregnancies, and we excluded mothers with major medical illness, including preeclampsia or chronic hypertension. From an original cohort of over 1,400 women, we matched preterm cases with term controls 1-to-1 by gestational age at the first blood sample, and then least difference in time between the two samples within the matched pairs. The result is 27 women in each group, 54 women total in the study. Next, we'll examine the most important results from this work. We found no significant difference in maternal serum allo levels between the term and preterm groups at these 2 time points investigated. Therefore, our findings do not support our hypothesis. This regression model shows little difference between the groups. Pink signifies term pregnancies and blue preterm pregnancies. Allo increased slightly more per week in term pregnancies, but it was negligible. We do start to see a slight divergence starting at about 27 weeks gestation. Here's where we had some exciting unanticipated findings. We found that preterm cases, seen here in blue, had significantly higher cortisol levels at the first blood sample and then significantly lower % change between samples. To summarize, we found no significant difference between maternal serum allo levels in term and preterm birth, and therefore our hypothesis is not supported. Further investigations using additional time points in pregnancy may be informative. Cortisol was significant, with preterm cases having significantly higher cortisol at their first blood sample and then significantly lower % change in cortisol between samples. This finding regarding cortisol may have important translational implications for clinical research, because if we can identify a woman at risk for preterm birth early in pregnancy, well, then we have an opportunity to intervene. This also builds on the literature regarding windows of developmental plasticity, in which environmental cues may be more or less influential depending on when they occur during gestation.
I'm excited about our cortisol findings. First, because they're congruent with the literature. But second, from a developmental perspective, cortisol may be a stress-related endocrine signal that triggers a hormonal cascade resulting in an abbreviated gestational period. In this sense, prenatal stress may be understood to accelerate maturation, similar to findings between prenatal stress and shortened telomere length as evidence for accelerated cellular aging. Accelerated maturation may be seen as a distinct phenotype, and from this perspective, certain preterm births may be a natural consequence of stress early in pregnancy, as a result of an ancient stress response system that's been selected for continually since it was present in the first vertebrates. Knowing that some women are disproportionately burdened with stressors coming into pregnancy and additionally disproportionately receiving inequitable access and care coming out of pregnancy, can this perspective of preterm birth as accelerated maturation help us illuminate, reduce, and even mitigate some of these persistent disparities that characterize preterm birth? I look forward to discussing these questions and thoughts and others with you at the live session. Thank you.